Asexual reproduction is a type of reproduction which does not involve the fusion of gametes or change in the number of chromosomes. The objective of this experiment is to introduce students to the asexual reproduction structure pattern in some fungi and use this criteria for their identification.
Saccharomyces cerevisiae was examined microscopically and it was observed that it has flat surface on top and smooth-looking shaped colony. Next, the texture of the yeast may be vary in cores which are moist, glistening, and dull, depends on the side of the plate. Secondly, Schizosaccharomyces pombe was examined and it has the same shape as Saccharomyces cerevisiae, which is flat surface and smooth colony on the top. The colony appearance texture is butyrose, which is it has the consistency of melted butter. The third culture that was examined microscopically is Candida albicans. It was shown that it has a venous shape of colony and butyrose colony appearance texture just like Schizosaccharomyces pombe. The color of the colony, however, differs from both of the cultures, which is deep red spots with background agar remain yellow. Epsidia. The shape of the culture is clearly can be seen as flat surface and smooth-looking colonies on the top. In addition, the colony appearance texture is moist, glistening, and dull. Aspergillus has the shape of globose ellipsoid, which is closed surface of which all plane cross sections are either ellipses or circles. The culture also has smooth and finely roughened colony appearance texture and pale brown roughened color of colony. Geotrichum. The top surface of the colony is flat and the colony appearance texture is dry and finely soot light with no reverse pigment. The color of colony is white to cream-like. Under microscopic examination, the reproduction structure of Saccharomyces cerevisiae takes place by budding. The protoplasm of vegetative cells swells up at one side in the form of bud. The nucleus undergoes mitotic division. Finally, the cells get detached and grow individually. Next is Schizosaccharomyces pombe, which is reproduction structure by binary fission. We can see a single vegetative cell forms two daughter cells of equal size. After nuclear migration, one at each side, partition wall forms almost in the halfway of the mother cell, and as such, two daughter cells are formed. Moreover, for Aspergillus, we can also clearly see that the mold is going through reproduction by blastic. In blastic division, the parent cell enlarges and a septum separates the enlarged portion into a daughter cell. This division may occur in differing arrangements. Last but not least, for Geotrichum, the reproduction structure sporogenesis pattern is stellic, just the same as Candida albicans. The initial spore differentiates after one or more septa has been delimited, and the spore differentiates from the whole cell. The initials of spore are delimited by septa produced after the fertile hyphae has ceased to develop apically.
Discuss the correlation between macroscopic features and the pattern of asexual reproduction in fungi. Describe the sporogenesis of each pattern: budding, binary fission, free cell formation, thallic, and blastic. Why do you think they possess different patterns of sporogenesis?
In conclusion, now students finally can understand and determine asexual reproductive structures or patterns in cultures of yeast and molds in order to identify it. That's all from us. Thank you for watching.